Okay, so this is the insertion of gaps. We were aligning these before, if you remember. And remember, we're doing bioinformatics evolution sort of stuff, okay? So remember that you can drop and create gaps, deletions, right, in your DNA. And so when we look at these in the reverse, we actually may need to insert gaps where deletions would have occurred, right? And so these insertion of gaps, they can allow the alignment method to compensate for insertion or deletions of nucleotides in the course of evolution. These may allow identities found in different alignments to be represented in one alignment. They can increase the complexity of sequence alignment. So there are scoring systems. You need to know how well each one scores, i.e. how likely it is to have occurred, right? And so the methods developed for the automatic alignment of our sequences will use various different scoring systems. This will enable the comparison of different alignments. This will introduce penalties for things like gaps. And it literally is like that. I use so many gaps, I get so many points off. This had a run of this length, I get this many points for that. Okay? The example of a scoring system, note that these things are highly contested models that are being published and upgraded all the time, okay? So I don't want to specifically give you a scoring system that I'm going to be like, this is the scoring system. Because in a year it probably won't be, okay? As long as they're doing their damn jobs. So the example of a scoring system would be something like plus 10 points for each identity, minus 25 points for each gap. This gap would be regardless of size. That actually makes a lot of sense if you think about it, because there is no reason that you would have to have a 1 base gap versus a 10 base gap, right? You could have that fragment broken off, okay? Right here, we have addition of gaps that are improving our alignment between the myoglobin and the alpha hemoglobin. So we had 38 matched amino acids, the average length of 147 residues. Our sequences are 25.9% identical. Yes, that is that 38 divided by 147, right? Okay. So the statistical significance of the alignments can be estimated by shuffling. And that's going to be it is possible that a grouping of sequence identities will occur by chance alone. And the significance of alignment can be assessed by randomly rearranging one sequence and determining the new alignment score. So what we can do here is, and this is why you would normally have more points for matching longer strings. If we were to shuffle and do random alignments, okay, that happens. You could get 4 random ones that are the same thing, right? Kind of like when they put typewriters in with chimpanzees, every now and then they actually form sentences, right? Okay. But if you test that on how likely it is to occur, you can estimate how likely it is that that would occur in this case. Okay? And that's what you're doing here, shuffling. So shuffling is going to be repeated many times. This will yield a histogram. If the original score is appreciably higher than the shuffled alignment scores, it's unlikely to have occurred by chance alone. Okay? It's a bit like what I do with your tests. If it's 4 answers, multiple choice exam, and you get 25%, how likely is it that it was just random guessing? Pretty likely, right? Okay. If you get appreciably higher than the possible 25% you could get, then it's probably not due to random guessing, right? Or appreciably lower. All right. So anyways, this is what we're seeing here, and we can see the alignment score for our match is much better than a random chance would give you. Okay? Notice, though, you can actually get pretty high alignment scores for random chances if you just get lucky, right? So distance evolutionary relationships can be detected through the use of substitution matrices. More sensitive scoring systems take into account the degree of similarity of amino acids. Conservation substitution is going to be where you replace one amino acid with one that is similar in size and chemical properties. This may have only minor effects on your protein structure and function. We had kind of talked about this already in codons, remember? Yeah. And that's the same thing. That's the same reasoning, right? Non-conservative substitution. This is going to replace one amino acid with one that is structurally dissimilar. Changes can also be classified by the fewest number of nucleotide changes needed for the amino acid change. Could you have in a binding site a change that goes from polar to nonpolar? It'd be very unlikely that that enzyme or protein would still work, right? If it's in a binding site. But could you have on a protein a change that goes from polar to nonpolar? Yeah, if it's just a chain that juts out into nowhere, right? Side chain, then it wouldn't really matter, would it? But it still is more likely that similar ones would get substituted with no problems, right? That statement is still true. So substitution matrices, or a substitution matrix, which is the singular, singular is matrix, multiple is matrices. Scoring system for the replacement of any amino acid with each of the other 19 amino acids. Large positive scores will correspond to frequent substitutions, ones that you could easily substitute with no real effect. Large negative scores will correspond to rare substitutions, ones that would have a significant effect on the local environment, right? Blossom 62, this is the commonly used substitution matrix. This scoring system detects homology between less obviously related sequences with greater sensitivity than otherwise you would with alternative methods. This is actually the substitution matrix right here, in case you're wondering. And this right here is the scoring. This would then be the amino acids that we would start with right there. And as you can see, we have different amino acids we could transform in. If it's the same one, the score is obviously going to be very high, right? And if it's different ones that are still pretty similar, the score will still be positive. Okay? Negative if they're very dissimilar. Is that making sense? Fantastic. Okay, so conservative and non-conservative substitutions will yield very different scores in your Blossom 62 matrix. So for example, we have the substitution of lysine for arginine. This would be a conservative substitution. They have similarities, right? And so it's a positive score. Notice that it is not a huge positive score. Maintaining the exact same one would be the biggest positive score, right? But it is still a significant positive score. The substitution, for example, lysine for tryptophan, this is non-conservative. These are very different functional groups, right? And so it would be assumed that on average, overall, any substitution like that would change how protein folding, it would change interactions, etc. The places where it would not change things would be less common than changing. Counting conservative substitutions actually reveals strong similarity between the alpha hemoglobin and the myoglobin here. So many of your different alpha hemoglobin and myoglobin are going to be conservative substitutions with scores that are greater than 0. And so we can actually see these here in the yellows. See that? Okay. So you got direct matches that would give you scores above 10, essentially. And then we have here substitutions that would be conservative, and those will give you positive scores still. Okay, so if we look at identities only, what we can see here is that this is our alignment score right here. Okay, and this is actually the one that is the one that actually shows up as Blossom being the more likely one. See that red one there? Which would not be the one that we'd actually take if we just did identities, would we? No. So if we take Blossom 62 as being closer to the real, the facts, then this one has the highest alignment score. And that's again because of the conservative substitutions. Okay? Okay, so this is your alignment of the human myoglobin and lupin Lego myoglobin. God. I really need to like start gargling before this class starts. Or not have a lecture before this and do this as the first lecture. So Lego myoglobin, excuse me. There we go. All right, does anybody have any questions about this matching? What they're looking at? No? What are the yellow ones? So the peach ones with the boxes are the ones that match. The yellow ones are the ones that are conservation changes, right? So they're similar, but not the same. Okay. And then the ones with nothing are non-conservative. They'd be negative scores on Blossom. So the rules of thumb for sequence analysis: for sequences longer than 100 amino acids, sequence identities will be greater than 25% or probably homologous. They're related somehow. Less than 15% are unlikely to indicate statistically significant similarity. Between 15% and 25% identical, they're going to require further analysis. They could be, they could not be. They're less promising to be homologous, okay? Or less promising to be different. That's what you're looking for. The lack of statistically significant degree of sequence similarity does not actually rule out homology, okay? You can have really drastic changes. I mean, what happens if an entire chromosome leg gets broken off? Or going from X to Y. If you had something like that evolutionary event happen again. That's a huge change, right? Yeah. So this is BLAST. This is basic local alignment search tool. This is a search method. It's going to be a procedure that's used to search a non-redundant database of all known sequences for homologous sequences. This is actually the site in case you want to go there and try your hand at this. Though without the software components to help you along with it, it's a bit of a slog. Okay? You have to kind of get used to, it used to be Perl scripting. I don't remember if it's still Perl scripting. But yeah, that really helps. Probably still is Perl. Perl's pretty used. Anyways, BLAST searches yield a list of sequence alignments. This is an estimate giving the likelihood each alignment occurred by chance. This is actually probably the thing you've actually seen before somewhere. Like them searching a database and this thing is related to what? In like sci-fi shows and stuff? Yeah. At least you're seeing a representation of it. Okay? Not always the real thing. Anyways, this is the related protein sequences because it's going to be rapidly identified using online BLAST search results. So here we have the name here of the homologous protein. This is what you would see in a BLAST search result. Here is the sequence ID right there. That's the length of it. That's the number of matches. And so here we see ranges. This would be the 252 to 920. Typically, you would have some ability to move back and forth through this range. Okay. I'm pretty sure this hasn't changed since your book was published, and they gave me this wonderful figure that I could deal with. Let me put it this way. My screen captures don't look as good. They did some massaging to this. This is the sequence here of queried protein. So here we have the query number here. This is the sequence right here of the homologous protein. And again, that's right there. And we can see the sequences and how they match up. And these are the differences right here. Okay? Okay. By differences, I mean differences blanks. See that it tells you the matches. And it tells you the ones that may be related. So we have an addition here. Okay. Again, the plus sign will be a conservative substitution here. Letter, again, is going to be the identity right here. Okay. Anybody have any questions? Fantastic. Okay. This tells you the frame, by the way. Here's the gaps, positives, identities. All of this, this right here is the number of sequences with your similarities expected by chance. In this case, are we expecting a lot of chance here? No. Can everybody read that number okay? 2 times e to the negative power of 25? Okay. Very unlikely to be by chance, right? Okay, so your blast searches with sequences from Haemophilus influenza, the first complete sequence of the genome of the free living organism, the bacteria Haemophilus influenzae, was reported in 1995. Your blast searches with 1,743 identified protein coding regions, or opened reading frames, or RFs is the abbreviation for that, open reading frames. That's useful, by the way, to know the abbreviations, because oftentimes it can only report abbreviations in these search results. Okay, you don't have room for anything else. Anyways, 1,007 ORFs, 58% were linked to some previously characterized protein of known function. 347 ORFs, again, that's the open reading frames, were linked to sequences in the database without a known function. These are hypothetical proteins. We may not have found them, or we may not know what the hell they do or what they're for. Okay? Oftentimes, they're things that are read off of DNA and we think they exist. Okay? 389 ORFs. These did not match any sequence present in the database at that time. Does that mean that it doesn't exist? No. Yeah. We're continuously adding new organisms, new DNA, new proteins all the time, right? Okay, so the examination of three-dimensional structure enhances our understanding of our evolutionary relationships. The tertiary structure is more closely associated with function than the primary structure. You all may have noticed this already from our proteins works that we've done so far, right? The tertiary structure is going to be more evolutionary conserved than is the primary structure. Again, we had mentioned that you can swap out amino acids in structural components fairly easy, right? And there's quite a bit of structural component, okay? However, can you change the tertiary structure of, say, a myoglobin very easily? No, that's going to cause you issues, right? What happens if we turn the myoglobin so it's a straight chain? Yeah, we're going to start getting things like sickle cell, right? Yeah, which is sickle cell's hemoglobin turning into fibers, right? Okay, and so right here we see some three-dimensional structure. There's a lot of similarity between the lupin legomoglobin and the human myoglobin, which is in the middle one right here, and of course, hemoglobin, which is the alpha chain in this case. Don't forget the hemoglobin has 4 subunits, but these are only 1 subunit ones, so it's better to compare the subunit to it. The human myoglobin is actually weakly detectable at the sequence level. The human alpha hemoglobin is not significant. It's only a 15% identity match with this. Okay? So the myoglobin seems like it's a lot more related, right? The actin, HSP70, are going to be paralogs. So our actin is a protein that's a major component of the cytoskeleton. Our heat shock protein 70, which is what that abbreviation is for, heat shock protein. That's the protein that assists the protein folding inside of cells. The proteins only have about 16% sequence identity, but they're paralogs based on their three-dimensional structure, meaning they have a relationship some way, right? Likely functional relationship, but they could have an evolutionary relationship, right? Even though they only have a 16. And so this sort of tells you, when you see this, low percentage relationship looks almost the same. This tells you should probably do some more work there, right? Yeah. There's more to discover here. Okay, so the regions and residues critical to protein function are going to be more strongly conserved than other residues. The sequence template, this is a map of the conserved residues that are structurally and functionally important and are common to a particular family of proteins. This allows detections of homologs that may not otherwise be apparent. Because again, like we mentioned, the things that are involved in the function of the protein, these need to be conserved, right? So things that cause the folding to be the way it is, or things that are in the binding sites, or things that are in regulation sites, right? Those change, the function changes, doesn't it? Okay. What if you change this amino acid right here? Is it probably going to have a big deal? No, the side chain is jutting out into solution, right? As long as it's something moderately polar-ish, probably not going to care, right? Okay. Even the hydrophobic one there probably wouldn't matter as long as we have other hydrophilic ones to offset, right? So we can do a lot of evolution on these sort of stuff. The stuff that actually binds things, that's where the evolution becomes more critical, right? So repeating motifs, these can be detected by aligning sequences with themselves. Many proteins have sets of 2 or more domains that are going to be similar to one another. These repeats can be detected by performing sequence alignments with the protein itself. And this is what we're looking at right here. So again, we're detecting these internal repeats using self-alignment. So this is the tatted box binding protein right here. And here we see the tatted box binding protein again. And so we slide this along each other and we can see a 40% identity match. We get them completely on each other, we see a 100% identity match. And then we keep sliding that across, and what we see is that we get a 40% again. And so these are the ones that have internal region similarity, and these are those repeating motifs that are showing up in this protein. Can you see that okay? Okay. I.e. things like alpha helices, and you can get more complicated ones like alpha helice turn, alpha helice, right? Or the phosphate loops, okay? That sort of thing. Okay, so the convergent evolution is going to illustrate the common solutions to biochemical challenge. Divergent evolution is a process by which proteins derived from common ancestor accumulate differences over time. They can potentially acquire new functions. The convergent evolution, on the other hand, is going to be the process by which very different evolutionary pathways lead to the same solution. Some of the examples, and very specific, would be serine proteases. Okay? Serine is incredibly important for many different metabolic and binding functions, right? And reaction functions. And so a serine protease is going to develop no matter what, basically. And it can develop multiple times convergently. So these are the mammalian chymotrypsin, and then we have the bacterial subtilicin. Again, these are going to be convergent evolution here. And so what you're seeing is ones that target this histidine here, right? And why is it important that we target it? Well, we have the catalytic triad histidine, right? That shows up everywhere. That's incredibly important that we can regulate, target, do things to it, right? So we get very similar structures, very similar functions, but it's pretty obvious if you start looking at them, there's a lot of divergences here other than the binding region, right? Okay, can everybody see that okay? Okay, so convergent evolution. So the comparison of the RNA sequences, these can be the source of insight into RNA secondary structures. Homologous RNA sequences can be studied to yield insights into evolutionary relationships and three-dimensional structures. And so this is the example right here that we can be given. So we have these RNA sequences, and we're studying them. And here we have different bacteria, and archaea, and eukarya. And what we can see is repeating patterns in these. And these tell us important relationships that exist among these organisms, things that have to be conserved highly because they're incredibly important. Okay? And so what do we notice about this that matches up to that 9 and 22? Why is that being conserved? So that's that hairpin loop, isn't it? In our RNA sequences. We don't have that. We don't stop the translation, do we? And so if we don't have that binding right there, we're in trouble, aren't we? Okay? And so that is conserved. Anytime that gets mutated, guess what happens? You die. Or at least you don't breed as much. Okay? Okay. So evolutionary trees can be constructed on the basis of sequence information. The evolutionary tree would be a branching diagram constructed from alignment sequences. The length of the branching connecting each pair of proteins is going to be proportional to the number of amino acid differences between sequences. This is just a different branching. Have you guys seen these evolutionary trees before with the protein matching? Rather than relationship due to, like, this has wings. No? Okay. That's fair. Okay, this is this right here, okay? So instead of having an organism, you actually have proteins. And this shows you how they're related to each other, i.e. the number of differences gives you a different branching. Can everybody say okay? Okay. Okay, and again, that's time millions of years. And then we have this. It's less related to hemoglobin than it is myoglobin, probably, right? The alpha one is more related to those than the hemoglobin beta might be. Okay? Or at least they diverged at a similar time. Anybody have any questions about that? No? Okay. It's just like evolutionary trees, but instead you're looking at proteins, right? Okay, these can be calibrated using fossil record data. Calibration occurs by comparing the deduced branch points with divergent times determined with the fossil record. The duplication leading to the two chains of hemoglobin appears to have occurred about 350 million years ago. This is supported by the fossil records on the divergence of jawless fish from bony fish, which was about 400 million years ago. And so those events likely occurred at similar time periods. Okay, this is the lamprey. This is a jawless fish. It is absolutely fucking nightmare-inducing. If you've ever seen one in person. Do you know how they exist? They attach to fish, and then they can grow up to, oh God, it was like 10 meters long. Yeah. And then they suck out their blood. Yeah. Yeah. Yeah, fucking terrifying. Nightmare-inducing, right? Anyways, the horizontal gene transfer events may explain unexpected branches of the evolutionary tree. Gladiaria sulfurea is a unicellular red algae that can thrive in extreme environments. These include things like temperatures up to 56 degrees Celsius, pH values between 0 and 4, in the presence of high concentration of toxic metals, so pretty nasty environments. This is it right here. Ooh, ah. See the wonderful, beautiful red color? There we go. Say hi. All right. So horizontal gene transfer. The horizontal gene transfer is the exchange of DNA between species that provide a selective advantage to the recipient, well characterized in prokaryotes. The eukaryote G. sulfurea contains genes more closely related to bacteria and archaea. These genes allow G. sulfurea to live in hostile environments, and that's why that's adapted that way. So the proteins that encode within the gene sulfurea genome have a high homology to prokaryotic sequences, which is kind of interesting, right? You may not have expected that. And so right here, we see the homology with the bacteria. Okay. These are the G. sulfurea. There we go. Anybody have any questions about that? No? Okay, fantastic. Okay. The reason you may not have expected that is that it's actually in this group right here. Anyways, that's why it's red here, by the way. See, that's not blue like the other bacteria, even though it has a high homology with them. Okay. So modern techniques make the experimental exploration of evolution possible. Two biochemical techniques allow the examination of evolution directly rather than by inference. We have the PCR, which how many times have we talked about that? 3 or 4? Anyways, you know PCR by now, right? Okay. So you probably get how this works. We get a DNA sample from something, it's highly degraded, we don't have a lot of it, right? Maybe from just a single hair that actually preserved. And so then we take that and we use PCR to amplify that gene. Do we get perfect copies? No, there's error in PCR, right? But on average, we'll get a pretty good copy. Okay, and our average should be pretty close to the original. Okay. We can synthesize highly diverse populations of molecules and selecting for biochemical properties. We can see what they would function like. Did anybody know that they're cloning a woolly mammoth? Yeah? Okay. In case you're wondering, yeah, they're cloning a woolly mammoth. They're going to bring it back. Ancient DNA can sometimes be amplified and sequenced. The complete genome sequences of a Neanderthal and a Denisovan have been obtained using DNA isolated from nearly 50,000-year-old fossils. And so right here we can see how closely they're related utilizing this DNA from those fossils, right? We can do that same sequencing matching that we had talked about before. Okay? Okay, neither are actually on the direct descent leading to Homo sapiens, right here. Though I must warn you, the science around Homo sapiens and our evolution is currently in a kind of flux state, okay? They found a much older site than any previously known site for Homo sapiens in Europe, rather than Africa. And yeah, so there's some crazy shit going on. And they keep finding that like different species that are hominids, we have fragments of their DNA in us somehow. So apparently we bred with just everything. Yeah, go figure, huh? Anyways, that's Homo sapiens for you. All right, so the molecular evolution, these can be examined experimentally. So for a population to evolve, 3 conditions have to be met. The generation of a diverse population, because if you have all brothers and sisters, that's not going anywhere, is it? Okay. The selection of members based on fitness criteria, the reproduction of that population to sequential generations. But by definition, you have to have reproduction in a population to sequential generations for it to still exist. Combinational chemistry or combinatorial chemistry, depending on which side of the pond you're on. The process by which millions of compounds are synthesized in a single process. This is used to synthesize a diverse population of nucleic acid molecules. We can then, basically this is a way of quickly trying out evolution and kind of like running an evolution simulation in real life. Okay? So we then use affinity chromatography. It'll be used to select molecules that bound ATP from a pool of RNA molecules. And so this is our RNA randomized RNA pool. We're going to apply the RNA pool to this column. This has an ATP affinity column. And so then we'll select these random ones that we synthesized in this column because they will stick. The stuff that doesn't stick doesn't have any affinity for ATP, does it? Okay. This is then the selective. We'll then add some loose ATP. This will help us elute the RNA that's stuck to ATP. And so then we can get versions of different RNA molecules that would have stuck to ATP, meaning they're biologically active in that, right? Okay. The molecules then bound to the ATP affinity column were replicated by reverse transcriptase into DNA using an error-prone reversion transcriptase to introduce additional diversity. So you caught that, right? That we intentionally did a bad job, right? Because that's what we're looking at. That's evolution, isn't it? Evolution is not, ooh, we're getting better. It's, whoops, fucked up. Oh, it benefited me. Awesome. That's evolution. Okay. So this is amplified then by PCR amplification, and it's then going to be transcribed back into RNA. What this does is it gives you the possible fuck-ups that actually make sense, right? Rather than just hammering it away with some chemical. After additional rounds of selection for ATP binding activity, molecules were then selected and sequenced. So this is actually a conserved RNA secondary structure. Right here we can see that 16 of 17 sequences that were selected formed this structure that we see right here, this red stuff. Each molecule bound ATP with dissociation constants of less than 50 micromolar. And so this is incredibly important to binding ATP and is a sequence that has to be conserved. As we can see, 16 out of 17 times, if it binds ATP, it has that. Okay? If we do the experimentation to molecular evolution, the aptamers, synthetic oligonucleotides that can specifically bind a ligand, in this case ATP, right? This will play a role in understanding the evolution, biotechnology, and medicine. It actually is kind of cool. It's not just an evolution thing. And so it's not just looking at history and past, right? We can also do things like selectively make something that would bind a specific enzyme we want to change the activity of, right? A specific substrate that is not currently binding and not being identified. Specific cancer agents, okay? We could bind cocaine. So then people, when they take cocaine, they don't get as much effect, right? Okay. All sorts of stuff. Anyways, here we can see that we basically get the same sort of loop we would have expected from binding an ATP in a physiological sense, right? Isn't that neat? That was just by random chance. Okay. Okay, anybody have any questions? Fantastic. All right, that is that.